In this video, we are going to take a look at some of the factors that affect enzyme activity. So we're going to take a closer look at enzyme concentration, substrate concentration, and remember the substrate is also known as the reactant. We will also take a look at how other factors like temperature, pH, and salinity may affect enzyme activity. And when we're talking about affecting enzyme activity, we're talking about the fact that enzymes work best in some situations as compared to other situations. So based on these factors, sometimes the reaction rate is increased and other times it is decreased. So let's start by talking about enzyme concentration. And concentration just refers to how much enzyme is present. Generally speaking, as the enzyme concentration increases, the reaction rate will also increase. And we see this in the first part of the graph here as the enzyme concentration goes up. So for example, if we start with 0 enzymes, and then let's say we add 1 enzyme, 2, 3, 4, etc., we'll see that as the enzyme concentration increases, the reaction rate is also increasing. But you'll notice that it reaches a point where that reaction starts to level off. So it does get to a point where adding more enzymes does not speed up the rate of the reaction. And we'll go into that a little bit more detail on the next page. Now let's take a look at the effect of substrate concentration on reaction rate. And remember that the substrate is also called the reactant. So this is the molecule that is reacting with the enzymes. So as the substrate concentration increases, the reaction rate also increases to a point, and then it starts to level off. You'll notice that this graph looks very similar to the graph comparing enzyme concentration to reaction rate. As we add more substrates initially, there are more collisions between enzymes and the substrate, and the reaction rate increases. But eventually, all the enzymes are saturated, meaning that all of them are busy breaking down substrates. So adding additional substrate will not speed up the reaction rate because all the enzymes are busy. Enzyme activity is also affected by temperature. And most enzymes have an optimum temperature at which they work the best. And so you can see this graph here that this is showing a temperature of 37 degrees, and this would be 37 degrees Celsius. And 37 degrees Celsius is normal body temperature. Or I should say normal human body temperature. And so this particular graph is showing you that this enzyme works the most efficient or works the best at that 37 degrees Celsius. Now, some enzymes are optimized to work better at different temperatures. So there's some enzymes that work really well in really hot environments and other enzymes that work really well in really cold environments. And so the graph would look very similar in that it would have this kind of curved shape to it, but the curve would maybe be in a different part of the graph, depending on where that enzyme is optimized to work. So typically what happens is if the temperature is too high, there's actually too much agitation. So molecules all have their own kinetic energy. So the enzymes and the substrates are all just kind of bouncing around and they're bouncing into each other. And as temperature increases, those molecules start moving around faster. And if they move around too fast, then when they bump into each other, instead of forming a bond, they actually kind of bump away from each other, and that bond is not formed and the enzyme can't work on the substrate. Sometimes if temperature is too high, the enzyme might actually become denatured. So remember that enzymes are proteins. And proteins have a three-dimensional shape, and that three-dimensional shape is really important. So we've talked a little bit about how enzymes and their substrates have this kind of lock and key or puzzle piece model, and that's because the enzyme has to match the shape of the substrate in order for it to work properly. So if you add too much temperature and it causes the three-dimensional shape of the protein to unravel, it's kind of like melting the key. So if you were to take the key to your door and melt it down, it's no longer going to fit in the keyhole and it's not going to unlock your door. If the temperature is too low, we kind of have the opposite problem in that the molecules actually slow down, so their kinetic energy slows down. And as these molecules are moving slower, there are not enough collisions or interactions between the enzyme and the substrate in order for the chemical reactions to be catalyzed as efficiently as possible. Enzyme activity is also affected by pH, which is a measure of acidity. So if the pH is not ideal, so if it's not the optimum range for the enzyme is not going to work very well. So they might lose their 3D shape. So once again, remember that shape is really important. If the shape of the enzyme changes, it can no longer do its job. And we call that being denatured. And so if the pH is not in the optimum range, then the active site of the enzyme and the substrate are not going to be able to meet up properly. So in humans, the enzymes that are in our blood typically work best in a pH environment of 6 to 8. And that's because the human blood pH is normally around 7.4. And many of our body fluids are also around that slightly basic kind of 7.4 pH. However, we do have different components in our body, or different compartments, if you will, that have different pHs. For example, we know that the stomach is really acidic, and that acidity is actually really important for enzyme activity because there's an enzyme called pepsin, and that pepsin only works in a really acidic environment. So without the acidity in the stomach, the pepsin cannot break down proteins in the stomach. Now, interestingly, trypsin is an enzyme found in the small intestine of humans, and it has an ideal pH of 8. And it's interesting because as the pepsin from the stomach moves into the pepsin actually becomes denatured, and then the trypsin actually becomes activated. So our body actually controls which enzymes are active by changing the pH environment of different parts of our body. Now, we didn't really talk about salinity, but salinity is also something that can affect enzyme activity. And by salinity, we mean basically saltiness. So, how much salt is present or how much ions are present. And even though we didn't talk about salinity specifically, it would have a similar curve as what we saw with enzyme activity and pH and temperature, where there would be an optimum range at which that enzyme would work best. So enzymes that work in the Dead Sea, for example, would work really well in a really saline or a really salty environment. Enzymes that work in the ocean work well in a salty environment. But enzymes that work well in fresh water would be denatured in the salt water of the ocean and vice versa. Now let's talk a little bit more about this word denature. So remember that proteins have a three-dimensional shape. Now remember that enzymes are mostly made up of proteins. There are some exceptions to that, but most enzymes are types of proteins. Now that three-dimensional shape really makes a difference. So if the shape changes, the enzyme is no longer able to match the substrate like puzzle pieces or like a lock and key, and the enzyme can no longer catalyze the reaction. So you can see this picture down here of a raw egg and a cooked egg. And this is a very clear example of what proteins are like when they are denatured. So an egg is made up of a lot of protein in both the yolk and the white. And when you apply heat to that egg, it cooks it. And you've probably noticed that there are a lot of properties to that egg that change. So the color changes, the texture changes, the taste changes. And all these changes are due to that protein being denatured by heat. Another example would be something like ceviche. If you've ever had ceviche, it's where they take usually like fish or shrimp or something along with some vegetables, and they soak it in an acid, usually lemon or lime juice. And the acidity denatures the proteins in the fish and kind of cooks it in a way. Other examples include things like pickling. So if you take cucumbers and you put them in a solution that's really salty and really acidic, it denatures the cucumber and it forms pickles. And so these are just all examples of how these proteins are changing when they're put into an environment that's not ideal. And in the case of, cooked eggs and ceviche and pickles, these are all yummy things that we eat. But if these were the enzymes in your body and they all of a sudden experienced heat that they weren't supposed to experience, or pH or salinity that was outside of their optimal range, they could stop working. And basically, when your enzymes stop working, you stop working. And so if your enzymes become denatured, then that can kill you. So this is one reason why homeostasis in living organisms is so important. So homeostasis ensures that those optimal ranges are maintained so that your enzymes do not become denatured. Now, one thing I want to point out, and this is kind of a pet peeve of mine, but enzymes cannot die. So you cannot kill and they cannot die. And that's because enzymes are not alive. Enzymes are just molecules, they're proteins. Proteins are not alive. They don't have all of the characteristics of living things. So you can't kill an enzyme, and an enzyme can't die. You can denature it though. So just that's a little bit of a pet peeve of mine when people use that terminology, because you can't kill something that's not alive to begin with.